The idea of using microfluidics, droplets, and capsule technologies for hormone regulation and body weight control belongs to the frontier, where engineering meets metabolism. At first, it may sound almost like science fiction. Tiny fluidic devices or capsules inside or outside the body that adjust how hormones are released, detected, or balanced. But when we look closer, the logic is clear. Hormones act at precise concentrations and times, and microfluidics is the science of delivering and controlling fluids in equally precise ways. In the human body, hormones like insulin, leptin, ghrelin, and cortisol orchestrate metabolism, appetite, and energy use. Obesity and metabolic disorders often arise not only from diet, but from the mistiming or misregulation of these chemical signals. Traditional treatments, pills, injections, or lifestyle changes tend to act in a coarse, bulk manner. But what if we could mimic the body's own feedback loops more faithfully? Microfluidics offers such precision by generating droplets and capsules that act as programmable hormone carriers. Droplet microfluidics can encapsulate hormones, peptides, or their mimics into uniform tiny packages. These droplets can be engineered to release their payload in response to pH, temperature, or even enzymatic signals present in specific tissues. For instance, a capsule could release satiety hormones only when glucose rises, damping appetite at exactly the right moment. Doesn't this sound more like the body's own rhythm than a blunt once-daily pill? Capsule technologies push this even further. Polymers, lipids, or hydrogels can form smart shells around droplets, controlling how fast and under what conditions hormones leak out. Some experimental systems already allow pulsatile release, bursts of hormone delivery that imitate natural cycles, like the pulses of insulin the pancreas produces after meals. If extended to weight control, one could imagine capsules releasing leptin to signal fullness, or blocking ghrelin at night when cravings typically spike. A fascinating and lesser-known possibility is closed-loop regulation. Microfluidic devices implanted under the skin or worn externally could sense hormone levels in real time, then trigger capsule release accordingly. Similar ideas are being tested in artificial pancreas systems for diabetes, but droplet-based microfluidics could miniaturize and generalize them. In effect, the body would gain an artificial feedback loop to help restore metabolic balance. The paradox here is that droplets, so fragile and tiny, might one day regulate some of the most powerful molecules in the body. Instead of thinking of them as lab curiosities, we can reframe them as synthetic endocrine glands, small, programmable factories that release exactly what is needed. Such technologies would not only target obesity, but could also help with conditions like thyroid imbalance, adrenal fatigue, or polycystic ovary syndrome, all disorders of hormone regulation. Of course, many challenges remain. Biocompatibility, immune response, long-term stability, and precise sensing of multiple hormones simultaneously are not yet solved. But the trajectory is clear. Microfluidics and capsule systems are shifting medicine from bulk interventions to fine-grained chemical control, mirroring how life itself regulates its most delicate processes. Hormones such as insulin, leptin, ghrelin, and cortisol are central regulators of appetite, metabolism, and fat storage. These molecules act as systemic signals, linking food intake to cellular energy balance. But dysregulation leads to obesity, diabetes, or metabolic syndrome. Current drug therapies often fail because they affect the entire body, not specific tissues, causing side effects. Whereas technologies that localize and fine-tune hormone release may revolutionize treatment, microfluidics and droplet systems offer unprecedented precision in delivering these regulators. Leptin injections can reduce appetite but often fail due to systemic resistance. It is like broadcasting instructions over a loudspeaker instead of whispering directly to the right worker. Microfluidic chips allow high-throughput testing of how hormones affect individual cells or tissues. By confining small volumes, these devices reduce reagent costs and accelerate experimental cycles, while droplet-based microfluidics can isolate single cells in nanoliter chambers, exposing them to precisely dosed hormones. Researchers can track responses such as insulin secretion or receptor activity in real time, accelerating discovery of new drugs and optimized hormone combinations for weight control. This also helps unravel the complexity of endocrine networks. Microfluidic screening has revealed how pancreatic B cells respond differently to glucose oscillations. It is like testing hundreds of lock-and-key fits in parallel rather than trying one at a time. Droplet microfluidics can generate uniform capsules containing hormones, peptides, or drugs. These capsules can be engineered for slow release, triggered release, or tissue-specific targeting, and encapsulation protects fragile hormones from degradation in circulation. It also allows tailoring release profiles to mimic natural circadian rhythms, which could mean regulating hunger or glucose with much finer control. In this way, technology mimics the body's own pulsatile secretion patterns. Encapsulated GLP-1 analogs are being explored to improve satiety and glucose control. It is like packing food in time-release containers that open exactly when needed. Advanced capsules could serve as implantable artificial glands. Microfluidic design ensures they sense environmental cues such as glucose and secrete hormones in response, turning them into closed-loop devices much like an artificial pancreas, but miniaturized. For obesity, capsules could detect elevated ghrelin and release satiety-promoting factors. Though the main challenge is ensuring biocompatibility and long-term function. Such biohybrid systems bridge biotechnology and living physiology. Bioartificial pancreas devices already use encapsulated cells to secrete insulin on demand. It is like installing a thermostat that not only reads temperature but releases warm or cool air automatically. Hormone action often depends on gradients, not just concentrations, and microfluidic platforms can reproduce such gradients with high precision to study how cells sense and respond. For weight regulation, this could clarify how appetite neurons detect leptin, or how fat cells respond to insulin waves. Gradient-on-a-chip devices allow simulation of rhythmic pulses resembling mealtime or fasting states, providing insights that guide design of therapies better matched to physiology. Microfluidic gradients of insulin have been shown to affect adipocyte glucose uptake patterns. It is like playing music with varying rhythms rather than a constant tone, producing richer effects. New capsules can be designed to respond to external triggers such as ultrasound, magnetic fields, or light, enabling on-demand release of hormones or inhibitors with non-invasive control. Patients or physicians could time release around meals or exercise, and microfluidics ensures capsule size and coating are optimized for reliable actuation. Such devices merge drug delivery with digital health, linking apps to body chemistry, and could transform weight management into a personalized, interactive process. Ultrasound-sensitive capsules have been tested to release insulin in diabetic models. It is like pressing a remote control button to deliver exactly the right message at the right moment. Microfluidics and capsules can pair with sensors that continuously monitor glucose, cortisol, or other biomarkers, creating a feedback loop where delivery is adjusted in real time. This reduces overdosing risks and adapts to lifestyle variability, while implantable sensors linked with microcapsules could automatically suppress hunger or boost metabolism. The result is a cybernetic metabolic system combining biology, chemistry, and electronics, representing the frontier of precision medicine for obesity. Closed-loop insulin pumps already approximate this logic in diabetes management. It is like having an autopilot for metabolism, adjusting course minute by minute. Altering hormonal balance carries profound risks, including psychological effects and systemic imbalances, so capsule or microfluidic therapies must be carefully tested for unintended outcomes such as reproductive changes or chronic inflammation. There is also the ethical question of enhancing metabolism for performance, not just treatment. While long-term implants raise issues of patient autonomy and control, regulatory frameworks will need to evolve to address these hybrid biotech systems, and public trust will depend on transparency and safety. Early appetite-suppressing drugs often fail due to dangerous cardiovascular side effects. It is like tuning an orchestra. If one instrument is adjusted too much, the whole symphony sounds wrong. The convergence of microfluidics, droplets, and capsules may create entirely new strategies for metabolic control. Instead of blunt systemic drugs, therapies could act locally, rhythmically, and adaptively, redefining obesity treatment from diet and exercise support to precise endocrine engineering. In the future, individuals might carry implantable capsules tuned to their physiology, much like customized hormone clocks. And combined with AI-driven personalization, this approach could transform global health. It shifts medicine toward regulation rather than correction. Personalized capsule-based devices could release hormones aligned with an individual's circadian rhythm. It is like having a tailor-made suit for metabolism, stitched precisely to your body's rhythm.